Hello everyone, welcome back to the channel Alpha Doctors, where we are studying the microbiology section, and today we are going to continue with the previous video. And in this episode, we are going to study the bacterial genetics. So bacterial genetics involves a few of the common mechanisms, including the transformation, the conjugation, and transduction. So transformation, as the name suggests, is transference of bacteria from the transference of the genetic material from one bacteria to another bacteria. So in this situation, one competent bacteria can bind and import short pieces of the environment naked bacterial chromosomal DNA. For example, when a bacteria dies, it leaves its genetic material in the naked form in the environment, as you can see here. And when a surrounding bacteria goes near this genetic material, if it's important, the bacteria can actually take that material and incorporate it into its own DNA and chromosome and genetics. So this process is known as transformation, and it's basically picking up of the genetic material, that is naked DNA, from the environment. So the transfer and expression of the newly transferred genes this process known as the transformation, and a feature it's a feature of many bacteria, especially the strep pneumo, H influenza type B, and Neisseria. Here is a short form that you can remember, and you will often see this in your first year, that is the Shin mnemonic. So the Shin stands for the strep pneumo, H stands for the H influenza, and N stands for Neisseria. So this is a mechanism in which there is basically addition of the deoxyribonuclease, which is an enzyme that degrades naked DNA and preventing transformation. For example, if we are culturing some growth, we're basically growing some organisms in different culture media, and we do not want them to take the DNA material of some other bacteria. So what we can basically do is we can just add this enzyme, that is the deoxyribonuclease, which will actually cleave this naked DNA, so there is no DNA available for the recipient bacteria to actually pick up. So this will inhibit the process of transformation. So this was about the transformation. Moving on, we have the conjugation. Alright, so next we have conjugation. What's conjugation? Conjugation is basically a transfer of genetic material from one bacteria to another bacteria with the help of some connecting molecule. So as in this situation, you can see this is one bacteria and which contains some extra genetic material. Let's call it plasmid, and this bacteria which does not contain, or it's basically the recipient bacteria that's going to uh, receive the extra genetic material from the its bacteria that's in its parallel placement. So this bacteria is actually the F negative cell, and the bacteria that is going to transfer genetic material is the F positive cell. So the F positive bacteria and F negative bacteria are connected through a junction. That junction is known as the pillars. So the basically pillars of this bacteria and pillars of this bacteria will be combined together, and this is and form a common plus that is known as the sex plus. So this sex plus will help with the transfer of genetic material from the F positive cell to the F negative cell, and after there's transfer of material, there's genetic information that will be common to both bacteria. So this is basically helpful in transfer or transference of some of the important pathogenic mechanisms of virulence factors such as antibiotic resistance among various bacteria. Then we have another important. For next up, we have the high frequency recombination. So what happens in this scenario, that the F positive bacteria that contains that extra piece of information, this actually incorporates this genetic material within its own chromosome. So once this plasmid is incorporated into the bacterial own genetic material, this bacteria is turned from F positive to the HFR. So what HFR stands for is the high frequency recombination cell. And when there is transfer of a leading part of the plasmid and few flanking chromosomal genes, that is this new genetic material from this bacteria, to its parallel bacteria, that is the recipient bacteria, there is basically a transfer of not only plasmid but the genetic material as well. What we need to remember the important distinction between the HFR and the F positive and F negative conjugation is that in this scenario, the cells that is the F negative will remain F negative at the end of conjugation, whereas during conjugation process in which there was transfer of plasmid material from one bacteria to the recipient bacteria, the cell that was originally F negative was converted into the F positive, whereas in this scenario where there is a transfer of the high frequency recombination material, there is no conversion of F positive bacteria, F negative bacteria into F positive bacteria. So conjugation is of two types. It's the transfer of plasmid or it's transfer of the genetic material, highly frequency recombinant material. Then the last process is the transduction. So our transduction is basically a packaging error. In transduction, the lytic phage, which is a bacteriophage, a virus, it infects the bacterium, leading to cleavage of bacterial DNA, and the parts of bacterial chromosomal DNA that are cleaved may become packaged into the phage capsid. And when this phage divides and infects the next progenies of different bacteria across the environment, it transfers this genetic material from the originally infected bacteria to the subsequent bacterial species, converting them to and giving them those unique characteristics from the parent bacteria. So this process is known as the generalized transduction. And what happens in the specialized transduction is that in this bacteriophage, basically that's known as lysogenic bacteriophage versus the lytic bacteriophage. In the generalized, the specialized contain the lysogenic bacteriophage. So what this lysogenic bacteriophage does, it infects the bacterium just like the lytic bacteriophage. The viral DNA inserts into the bacterial chromosome, and when phage DNA is excised, that is, the flanking bacterial genes may be excised with it. So the DNA that is packaged into the phage capsid and it can infect in the bacterium. But in this process, there is no death of the so in this there is no death of bacteria. What happens in this, that is the new phage from the lysed cell can infect other bacteria, and genes that are formed, they'll be different from the donor and the recipient. So the genes for the following five bacterial toxins are encoded via the lysogenic phage. These include the M group A Streptococcus erythrogenic toxin, the botulism toxin, the cholera toxin, the diphtheria toxin, and the Shiga toxin. So these five toxins are important to remember that are transferred through the lysogenic phage system. Then last and the uncommon mm, transfer of genetic material happens with transposition. So transposition is basically jumping process that involves a transposon, which is a specialized segment of DNA which can copy and excise itself and then insert into the same DNA molecule or an unrelated DNA molecule, such as the plasmid chromosome of an other bacteria, and it is critical in creating plasmids with multiple drug resistance and transfer across species lines, such as the transfer of when a mutation from Enterococcus to the Staph aureus, which provides its resistance to the vancomycin. Then and ending our session with some main features of the exotoxins and endotoxins. The exotoxins are the characteristic features of certain gram-positive and gram-negative bacteria. What is endotoxins is it is searched for the gram-negative bacteria only. So the exotoxins are released from the cell, whereas endotoxin is embedded in the cell and it is not released until the bacteria dies. Exotoxins are usually polypeptides, whereas endotoxins are composed of the lipopolysaccharide, and it is basically make the basic structure of the DNA cell, DNA cell membrane. And the location for production of exotoxins is within the plasmid or bacteriophage, versus the endotoxins are synthesized by the bacterial on chromosome. And the toxicity is high for the exotoxins, that is, they require a high fatal dose, whereas the endotoxins does not require a high fatal dose, versus they require the low fatal dose. Clinical effects of exotoxins are various, as we will study in our subsequent sections, and it affects via various modes of action, inducing high antibody titers called antitoxins. They can also be used in vaccines, and they are rapidly destroyed at 60 degrees centigrade except the Staphylococcus enterotoxin and E. coli heat-stable toxins. And the treatment of exotoxin is dependent upon the specific toxin that is causing the pathogenicity. For example, the tetanus toxin can neutralize the anti-tetanus immunoglobulins and like pest botulism, diphtheria, and cholera. So the endotoxins produce a common effect in the pathogenesis, that is the fever, shock, which includes hypotension and disseminated intravascular coagulation via the activation of TNF, interleukin-1 and interleukin-6. They are poorly antigenic, so there is no antibodies produced against the endotoxoids are formed, and there is no vaccine available. These endotoxins can remain stable at 100 degrees centigrade for 1 hour. And the typical diseases that are caused by the endotoxins include the meningococcemia and sepsis by various gram-negative routes. So this was all about the basic bacteriology and genetics. In our next section, we will study some detail about the bacteria that produce the exotoxins and their characteristic features. If you have any questions so far, you can ask in the comment section. And if you like the video, then please make sure to like, share, and subscribe the video. I hope you enjoyed so far. Thank you so much.